Hello, everyone. My name is Kenneth Witwer, and I'm recording this brief presentation today to share with you the results of a survey that we've recently completed for the Extracellular Vesicle Club. The Extracellular Vesicle Club is a journal and research club that meets weekly. It's available to everyone around the world, and it was converted from a monthly journal club that we held locally at the Johns Hopkins University prior to the COVID-19 pandemic. As we're coming up on a year of holding this weekly EV Journal Club and Research Club, we decided to take a questionnaire to the community to find out from you, from the EV research community, what you would like in this weekly EV club. This is the survey that we sent out. It was called "What's Next for Extracellular Vesicle Club? You Decide." This survey was held for 3 days at the end of February of 2021, and we gathered responses by posting links on Twitter, on LinkedIn, on the Facebook groups for ICEV and exosomes and microvesicles. And then we also emailed this survey to our core mailing list. We received 267 total responses. Where were people logging in from around the world? Well, we had 119 responses from the Europe Africa chapter of ICEV. We had 131 responses from the Americas. And then we also had 17 responses from Asia Pacific. So how are these broken down? The Pacific region had 7 responses. Asia would be about 6 respondents. And then we had 4 people who are coming from the Middle East. In Europe and Africa, we had 117 responses from Europe and 2 responses from Africa. And then in the Americas, we had 118 responses from the North American region and 13 responses from the South American region. Our first question was this one. Please select the best answer. I have ... And of course, the best answer of all was the one that referred back to the movie called Fight Club. So we said the first rule of EV Club. And that answer was no intention of disclosing if I have or have not attended EV Club. So we had at least 10 people who are fans of that movie and selected that answer. And I love you guys all. I love everybody, but I especially will keep these folks in mind in the future. So thanks for playing along. But the other responses that we had were there were a few people who had not attended EV Club, but were interested in signing up. And then most of our respondents had actually attended some or most EV clubs. The second question that we asked was which of the EV Club formats might be most useful to the community. 90% of our events so far have been journal club events. And indeed, 84.6% of respondents said that they would like to have journal club style formats. But we also saw that there was a lot of interest in some other formats, including research talks with unpublished data, that was 64%, and then debates or discussions on a central question or topic, which came in at around 65%. So what have we done so far? As I said, about 90% of our clubs have been journal clubs. We've had about 9% research or overview clubs. And then we did have one debate session that was a hybrid event. It was a journal club followed by a debate style discussion. 6% of people also gave an other answer. And so what were these other answers? Fortunately, nobody said interpretive dance, so they followed the instructions. But the other formats, of the 16 total that requested these other formats, 11 of them were about methods, techniques, and best practices. So they wanted to learn more about specific techniques or about specific approaches to EV research. One of these was also a request for a session on introducing the various databases that are out there. We also had some suggestions about looking at regulatory guidelines, of finding cooperations, and one person asked for maybe shorter journal clubs, but with more discussion. And in most of our weeks where we have a journal club, we have about a 30 or 40-minute presentation, followed by discussion to round out the hour. For the journal club formats in question 3, what do you prefer as far as presentation? 31.5%, so they didn't really have a preference here, but about 60% of people, almost 60%, said that they preferred if the author or the authors present their own work, whereas a minority of individuals who responded said that they would prefer to have someone who is not an author presenting the work. What have we had so far? Well, so far, of our journal club events, we've had 30 where authors are presenting their own work. We've had 6 where others present, but the authors are present on the call and usually participate in the discussion. And then we had at least 5, I think, where somebody else presented and the authors either were not present or didn't participate. Question 4, and this is very important for a global event. When do we hold this journal club? So 10% of you said that you're such fans that you would join no matter the time, day or night. And I think you people maybe need to calm down a little bit, but I appreciate the support. We had almost 30% saying that they preferred a time during the business day in the Americas. Also, more than 25% said that they preferred a time during the business day in Europe or Africa. In Asia, unfortunately, we didn't have very much response from the Asia Pacific region, as you could see from the world map. And there were only 4 people who said that they preferred to have a time during the business day in Asia Pacific, and that was just 1.5% of our responses. And more than a third said that they preferred to have an alternating schedule, so a schedule that would accommodate multiple time zones, I should say, depending on the time that's selected for each individual week. In question 5, we introduced another format that we've been talking about, and this would be something that we call the EXTRAtalks. So these are not full-length clubs, but they would be an opportunity for a group to present their work, likely a preprint or a recently published manuscript, in 5 minutes. So this would probably be about 5 minutes where you record something on Zoom or some other platform, and then you send the recording. And we could post that on our YouTube channel, Extracellular Vesicle Club. And we could potentially even play some of these at the beginning or at the end of other regularly scheduled events. So we asked, is this new format of interest to you or your group? And we had about 21% of the responses saying, yes, we actually have something now where we would like to record this and present it. 73% said they would likely have something in the future where they would be interested in this format. And then I'm really curious about the 17 people who said, no thanks, the less publicity for our manuscripts, the better. So thank you for your honesty and candor, and good luck to you. Thank you. Question 5 was actually a free format answer question, but it was mandatory. So everybody who took this survey, to be able to complete the survey, they had to enter something. And of those 267 free-form answers, I was actually quite impressed that most people gave very thoughtful answers. There were only maybe 5 or 6 answers that could not be interpreted or were non-answers. One of my favorites, actually, and I think this is a big controversy here, was, will we ever see clean-shaven Ken again? And I don't have an answer for you, but maybe I will in the near future. But the question was, in your view, what is the most interesting question or controversy facing EV science? And as I looked through these 267 responses, and frankly, there was a lot to read, I realized that we could break these answers down into several categories. These categories were, first of all, biogenesis and cargo sorting. So we're going to look at some of the specific responses. I'm going to read you some of those in a minute, but I want to go through these different categories. So biogenesis and cargo sorting. Then we had heterogeneity and categorization. EVs from specific cells, subtypes of EVs. How do we classify these things? How do we separate them from each other? Then we had the question of what happens when an EV reaches its destination, its target cell. What is involved in targeting? What's involved in that cell interaction, uptake, cargo delivery, and distribution? We had quite a few responses that had to do with physiologic roles and mechanisms, such as stoichiometry was one thing that came up frequently. But this was a very broad category. And I should mention, I also included some answers in more than one category. We had some interest, a minority of responses, but there was definitely a strong interest in cross-organism and non-mammalian EVs, so cross-organism communication. And this included some questions on viruses and EVs, and how, let's say, envelope viruses relate to EVs. Many, actually the biggest category and a very wide catch-all category that I assigned was on rigor and reproducibility in methodology. So people had questions about those separation techniques, about how we best characterize, how we can manipulate EVs, even our experimental design. So kind of following along with the different categories in the MySub paper. And then there were the questions or the controversies that related to translating EVs into the clinic. How can we use EVs as biomarkers, and can we use EVs as therapies? What are the questions around the efficacy of EV treatments and the regulation, the regulatory affairs? So let me take you through some of the questions that came up, some of the individual answers. This is just, first of all, a little word cloud that shows you some of the more frequently used words in these responses. And so you can see here the interest in the different methodologies, but also in what we're ultimately going to do with our EVs, which of course was the most represented word. The first recurring set of themes that I'm going to mention here is loading and heterogeneity. So what controls the sorting of cargo into EVs, and how can we control that? Quite a large number of responses posed this question. And it was protein and RNA that seemed the most popular cargoes, but some people talked about DNA as well. There is a lot of interest, obviously, in heterogeneity of EVs. So we had a question or a statement: tremendous heterogeneity of EVs and body fluids demands that we define and characterize EV subsets, including origin, phenotype, and above all, functions in health and disease. Another respondent said that until reliable single vesicle characterization and content profiling is a reality, we will have to cope with mutually exclusive claims about EV content, markers, and function. And I found this to be a particularly insightful comment because it shows us the limits, or it reflects the limits of bulk characterization of EVs and how we are now trying to move into single EV characterization to really capture the heterogeneity that is out there. And there was an emphasis on the rigor and reproducibility that we're trying to introduce into the field. Someone said increasing the standardization of EV classification and isolation methods, labeling methods, and another person said in an ideal world, every group would follow the MySev guidelines. Another recurring theme was about delivery and stoichiometry. So how is EV content transferred into target cells upon active uptake? How does the cell turn trafficking pathways on and off? How are EV cargoes delivered again into cell cytosol? And how do rare EV RNAs elicit such profound effects? So we've seen reports of very strong effects, but on the other hand, we also see reports of very little RNA per individual EV. Along those same lines, how are EVs present at high enough concentrations in blood to alter cellular function between distal organs? Or are most EV functions limited to local interactions? And to sum all of this up, I think, was the question, what are the in vivo realities? What role do endogenous EVs play in physiology and pathophysiology? Okay, so moving on to how EVs might be used in therapeutics, there were quite a few questions, quite a few controversy questions, I guess, that were raised here about the EV itself versus its maybe external interactions. Maybe the corona of the EV, the protein corona, maybe other particles that EVs associate with, and maybe co-isolates, so things that are not EVs, not even associated with the EVs, but will come along in our different separation methods for the ride. So the EV versus the secretome, in other words, is there any there? One person asked. Are EVs really the key component of a cell release rate? Someone said EVs, lipoprotein particles, ribonucleoproteins, and single or aggregated proteins can all show up on techniques like NTA, nanoparticle tracking analysis, and in other assays, and they may all have important roles. So what about the internal versus the external cargo in therapeutic efficacy? And even the question of whether we need to have uptake at all. So EVs have active surface molecules which can bind cell surface receptors. Are we looking into this enough as opposed to focusing just on the delivery of contents of EVs into the cytoplasm of a recipient cell? Someone asked, so why do companies insist on using the term exosome to refer to all contents of cell culture supernatant? And I would say this isn't just companies. We also have quite a few labs that are doing this, where there will be particular cell type. You take the supernatant from that cell type, and you might do a very crude fractionation, but certainly not an EV purification. And then anything that is in there is assumed to be EV. So it's a good question, I think. Someone said, we're soon to be hit with so many EV therapeutics that all basically do the same thing. So what is that thing? Is it trophic support? Is it signaling? Is it cargo delivery? Can we grind up or shake or electrify cells, or do we really need to have those EVs that are produced, quote unquote, naturally from cells? And then there were questions about EVs and viruses, as I mentioned before. Okay, so question 7 finally was an optional question, but people could give an answer of their favorite EV paper or preprint that's appeared in the last year or so. So we had 149 answers to this one, and some very interesting papers. And there were multiple nominations for these papers that are shown here. It turns out that most of them have actually been featured in EV Club already, but there were a few that were newer that have not been featured in EV Journal Club, and they include a recent publication in Molecular Therapy by Dooley and colleagues, some new engineering methods for EV loading. There's also a comprehensive evaluation of methods for small EV separation from different sources, and that just appeared in JEV. And then we also have one on tracing. So how can we look at biodistribution in vivo? And then finally, there was one that got 2 votes for interneuronal exchange of synaptobrevin via EVs. So these are perhaps some papers that we'll try to feature in the near future. But then there were many other papers that were nominated here. Some of them have been covered in EV Journal Club, some have not. I just want to mention too that there were some much older papers that were mentioned. So even though we asked for papers in the last year or two, there were some older papers that were mentioned, I think going back as far as 2005. So someone had nominated a paper from 2005 that talked about EV engineering. So some of the papers that had 2 or more nominations, I'd like to go through some of these because we had some very good journal clubs on these. And so in each of these cases, I'm going to show you the journal club slides, say just a couple of things about the papers, and then show you also in red here how many logins we had. So we have averaged about 130 logins, unique logins for each journal club. So I think reflecting a lot of interest around the world in hearing about some of these new papers. So the top vote getter was Hoshino et al. Cell 2020. And it was in this paper that came out of the Leiden lab with collaborators, they showed that there were extracellular particle populations, which included EVs, that could be separated by differential ultracentrifugation and then profiled. So they looked at these EV-containing EP fractions in different cancers and found some interesting results on the, for example, the surface protein markers that could be found in these different EV populations. Then we come to, there were several papers that had 3 nominations, and one of these was from Model and Berger. And so Ferreira Berger presented this in 2020. And this was a bioarchive preprint on how EV preparations can actually differ in their potency quite a bit from batch to batch. This makes it very clear that we need to be sure that we characterize our EVs very well, and ideally also potency test them before we put them into a preclinical study or into human patients. Then there was a paper on the very large vesicles termed exofers that can be released by some cell types. And here we were seeing that these large vesicles can actually transfer cargo and contribute to mitochondrial homeostasis in the heart. Next was a paper from Melissa Weaver's lab on a live cell reporter of exosome secretion and uptake, and how these EVs that are released can have paracrine and autocrine effects in cell migration. This was one of our, I think this was our first collaboration with the Student Network on EVs, or SNEV. And so this is a regular feature now of the EV Club, where on the third Wednesday of each month, we will have a collaboration with SNEV. We had a very interesting paper, I think, that asked one of those questions that had come up a lot in the controversies section of our survey. And this was by Tom Whitaker. And so he presented his work on EVs and other components of the releasate. So what is responsible for the effects of MSCs? He was looking in a particular system and found that many of the effects were due to co-isolated components like protein factors that were not necessarily associated with EVs at all. We had one, Bahara Hossein Khani presented on subpopulations of EVs. So she was looking at different populations that can be obtained by ultracentrifugation, including ICAM positive EVs, and how they would contribute to inflammation and monocyte migration. One of our first journal clubs, it was actually our second journal club in the series, was presented by Tom Dredonks, and he presented the work from Jay Debnath's lab by Leidel et al. On autophagy and extracellular vesicles. And specifically, we're looking at the loading of RNA binding proteins and how that's related to LC3. Unfortunately, this session was Zoom bombed. So we had a horrific Zoom bombing incident. Some of you may have seen that. And we actually had to stop the journal club and resume it again a few days later. So most people came back, though, for the second session, including the authors, and we had a really nice discussion. This is also one of our most watched videos on the YouTube channel. We also had a presentation from P.S. Silyander on extracellular vesicles from human blood products and how they are carriers of extracellular vesicle cargo. So these authors were also looking at the protein corona of EVs. And now I'm just going to show you a few of the papers that were also nominated and that were covered in EV Journal Club. So here we have a recent presentation by Raphael Leblanc from Pascal Zimmermann's group on inhibiting syntenin and what that does to cargo. We had a talk on nanoplasmonic approaches from Randy Carney. Marina Kretic talked about membrane binding peptides for EV on-chip analysis. We had a very, I think, a very popular session from Andreas Zietzer, who told us about a particular mechanism whereby RNPU can regulate the sorting of a specific microRNA into EVs. We also had a presentation by Michael Paulides on a paper from the Lee lab at Ohio State on how you can load EVs with cargo after they've been purified. Juan Pablo Tosar talked about his work on extracellular tRNA fragments. We had a report from Masa Somia on real-time luminescence assays for cytoplasmic cargo delivery. So how can we detect if an EV cargo is taken up? And this was a rather interesting journal club too, because they didn't find a lot of uptake. And so there were questions about how common cargo delivery truly is. We had a very exciting preprint that was presented by the author Alessio Lana on telomere transfer by EVs. Alvin Tu of University of Ottawa shared his recent comprehensive review on MSC-derived EVs. We had a report on synchronized Raleigh and Raman scattering from Agustin Anciso Martinez. Clotilde Terry's lab joined us as Aaron Scott presented a study on EVs that are released by HeLa cells and how there's actually more ectosomes than exosomes that are released under the conditions that they used. So looking at release of certain tetraspanins. Then we had a kind of hybrid journal club where we presented some work of the Rigor and Standardization Subcommittee of ISEV with Juan Falcon and Rink Newland. And this was quite a popular session with a lot of questions about what ISEV is doing to encourage rigor and reproducibility in EV research. And finally, I just want to mention our most popular, our most followed event. This was a club on EV microRNAs, and we asked, are they wimps or are they warriors? Manuel Albanese presented his preprint on how there's very little microRNA cargo of EVs and how they don't get into cells at large numbers. We had a little bit of a debate here between Manuel and Michiel Pechtel, who has been working on microRNAs and EVs for quite some time. As long as anybody in the field, really. And so we had a very good discussion between the two of them and also with the audience. And this is also our most watched event on the YouTube channel. So where does that leave us? What is really next for the Extracellular Vesicle Club? What I see coming up based on the results of this survey is that we want to introduce some methods sections. So where we talk about some widely used methods, maybe bring in a few presenters to talk about their approaches to those methods, and then have an extensive discussion about the method, about how it can be improved, about alternatives. We can also have other types of discussion sessions, so maybe sort of like the microRNA debate, but with a panel and moderator format. And we could address hot topics and questions and controversies here. I think we've done some of these types of things at the ISEV 2020 virtual meeting, at the ISEV Infectious Diseases meeting in January of 2021. We can certainly implement this type of a format for the EV Club. The EXTRAtalk format of 5-minute videos also seem to be of large interest to the community, so we will look into how we can best set that up. And finally, I think you're going to see a lot more guest moderators in the near future, so some new faces, not just my face up there every week. We've already had the collaboration with the Student Network on EVs, and we will continue that. And we will also seek to have more diversity of moderators as we recruit more people for the upcoming sessions. Well, that's all that I really had to say today. I hope you're all doing well, and I look forward to seeing you at a future EV Club.